Good morning, everybody. Welcome back. Happy back from the break day or whatever you want to call it. Holy smokes, I haven't had to talk out loud like this in a very long time. Hi, everyone. Welcome back. Good morning, Vicky, Colin, Jessica, Katie, Valeria, Stanley, Maha, Sarah, Olivia, Rocco. Welcome back, folks. Welcome back. Good morning, Ava. My break was good. Amina, good morning. My break was good. Yes, the break I found, I don't know if you all found this as well, but with COVID, it didn't really feel like too much of a vacation because the normal day that I have is like I'm at home. And then my break was I'm at home. So it was like kind of the same. And again, right, I wasn't doing this, which again, I don't know if you all can tell, I quite like teaching. So this is like a pretty solid part of my day that I did not have for a week. So it was a little bit like, what am I going to do? And so, I read a lot of books, played some video games, kind of the same jam, just different toast. So it was a little bit underwhelming, if I'm being honest, but it was nice to have a bit of a relax and not look at my emails every day and correspond with a bunch of people and all that jazz. So yeah, and the weather was so cold and rainy too. So exactly. Great point, Ava. Yes, I agree. It was not great. Dean, Daniela, Matthew, good morning, everyone. Welcome back.
Good morning, Mr. Kiedis.
Hey, Matthew, how's it going? I just read a lot, played some video games, went for a bike ride or two. I got a new bike, so that was nice. And yeah, I promised myself I would sleep in, and I ended up waking up earlier than you. Vicky, I always wake up at the same time pretty much every day. I think there was 1 or 2 days I slept in till about 6:37, but otherwise I'm up always at the same time. The rain is very relaxing for me as well, Rose. I like rainy days, but it would have been nice to to get out. 6:30 to 7 I would sleep in. Yeah, 6:30 and 7 o'clock in the morning I would wake up. 4 o'clock, holy smokes. Early. Yeah, there was a few days. Oh, PM. Never mind. Oh dear. You had to wake up at work for 4 a.m. That's early. Holy smokes. Holy. That is quite sad. I used to have to get there. I would quit. Yeah, that's a tough sell, but I hope you enjoy your job at least. My hope is that you enjoy it. They should give us 2 weeks off. Yes, it would be nice. I agree. 1 week is kind of tricky because it's not really enough time to get into a groove of being off. And right as I started to get used to getting off or getting up early, being off, now I'm back, right? So now you're all back after just kind of getting used to it. I know, I think 2 weeks would be nicer if the pandemic wasn't a real thing, right? So, but I digress. It's a coffee shop, so it has to open early. Yeah, that's it. Yeah, we barely got 1 week. I know, the week went by very quickly for me. All right, folks. So I got everyone in who I think is going to be here for now. I'll do attendance in about a minute or two. And then we'll kind of talk a little bit about what our plan is for today because I want to get Classroom opened up. If you saw, I made a post to our classroom with regards to our Unit 3. We start evolution kind of this week. So I'll bring up the schedule. I'll talk a little bit about the plan for this, the next 3 weeks, which is about how long our unit will be. I'm really trying to downshift this unit a little bit in terms of the amount of work and also in terms of the assessments. And I'll talk about that in just a second because I started marking, I'll be honest with you, I started marking 1 or 2 of the assignments and then I stopped because I'm I'm on break. I don't really want to inundate myself with too much work right now. I want to relax a little bit. So I took my break a little bit more seriously and I only really got through 1 or 2 assessments. And then I realized I can't really mark them fully because I need the second part before I finish marking it. So it kind of defeated the purpose of starting to mark it. How many more units are left? So we have evolution, and then we have human anatomy, and then plant anatomy. So 3 units left. This unit's going to take us right to about middle of May, early May, sorry. And then plants will be all of May and a bit of June. And then the last 3 weeks of June, right until the end, will be on plants. Yeah, evolution's only about 3 weeks. Plants is only about 3 weeks. And then animal systems is about 4.5. Yeah, we have 5 units. Yeah. I was just thinking the other day, like, why would they give us such a big project to mark over the break? Yeah. I'm like, yeah. Oh my God, April is almost over. It's technically spring. It is technically spring. I know. This is the first, I don't know about you, but it's like quite nice in my area today. Spring is definitely in the air. It's quite warm, actually. I gotta turn my fan on. Sorry if it makes a bunch of noise. Turn up my brightness. All right. So let me hold on a second. I figured I was allergic to pineapple over the break. Holy smokes, Daniela. Well, that's not good. Pin. All right. Where am I? Holy smokes. There's like a beam of sunlight right on half of my face. Honestly, Daniela, I had that allergy. I used to be allergic to every single tree fruit that existed. So if it grew on a tree, I used to be allergic to it. I've since grown out of it, but I still have, my body still freaks out from time to time. And I get hives if I have like apples or pears or something like that. But it's I used to have a very similar allergy. I know. I just bought a pineapple. Alrighty, let's take a look. Let me share my screen with you. Holy smokes, they're in season right now. Yeah, I know. It's I can't wait. We just got it. It's going to be ripe in about a day or two. So really looking forward to cutting into that pineapple. Sorry you can't enjoy pineapple anymore, Danielle. That's a bit of a bummer for you. All right, let me just make sure everyone is in that's here. I will load up Teach Assist. God, I haven't had to open up Teach Assist in ages. Emails. I should probably open those up too. Alrighty. Are we starting evolution today or just like a catch-up day? I'll show you what our plan is for today. I'll open up our schedule. So as you can see here, my little welcome back. We have our unit outline as well as the folder. So as everything before, I shared the folder with you, and everything we'll need for this unit is in this folder. The only thing I haven't posted that you'll need is the lab, which I'll talk about in just a second. So just make sure you can access these files. Make sure that they open up for you. I uploaded them directly from my computer. So my hope is that the Word documents will open up as Google Docs for you. If there's any problems with those files, please take a look at it. And yeah, we don't actually, like I said, we don't technically start Unit 3 proper today. I'm going to go over the schedule in just a bit, but you will need the stuff from the last 2 lessons from Unit 2. We're kind of doing a little self-directed / I'm going to talk about some cool things that are going to be important. So I'll touch more on that in just a second with regards to what you'll need, but I'll show you that once we finish going over the schedule. So the schedule, today is Monday, April 19th, and we are technically starting Unit 3 Evolution. So I am going to kind of do a little brief recap on the big important things that we will need for Unit 3, and I'll connect them to some of the ideas. We don't start the first lesson for Unit 3 until tomorrow. So it's not the end of the world. Today's a bit more of a relaxed day. First day back, I really want to try to pump the brakes, or at least kind of like relax a little bit and, you know, really try to talk about things in the context of what we'll need to do for the next couple of weeks. So as you can see, I've given you a work period on Friday to do any finishing up for your assignment. Remember that the assignment part 2 for Unit 2 is due at midnight. So if you did not do any work over the break, that's perfectly fine. I'm giving you this week as well as a full work day kind of today and then on Friday to kind of, you know, get some of that done and dusted. Some of you have submitted already. Thank you so much. I saw some of those. I'm going to try my best to mark them over the next 2 or 3 days, and then I'll release your mark at the end of the week when the assignment is due. Do we have to show our face? Yes. A couple of people reached out to me over the break saying, oh, my camera's not working. And I was like, okay, that's fine. But like, please let me know this earlier, right? Like the expectation is that you're on camera explaining so I can see you explain. If you're telling me like a week before it's due, that's fine. But it's just a question of, you know, if I can't see the understanding, I'm just going to listen. And if I can't hear what you're saying or what you're saying doesn't make sense, and I can't see your face comprehending, like some people can say some stuff that kind of makes sense, but when I see you explain it, I see that you know what you're saying, and that allows for me to make those contextual connections. If I don't see that, I'm just going to hear it, and I'm just going to mark it based on what I hear. And if you make any flubs or mistakes, and I don't see any of those understandings in your facial expression and confidence, then I'm just going to mark it as is. So, you know, please make sure that you know, that you have your camera on if you can, and we'll make do with what we got. All right, so that's our first week. And then I'm giving you your assignment on Tuesday of next week, April 27th. And then as you see, we have 2 whole house days on Wednesday and Thursday. I'm also going to give these as days for you to work on your lab. But the reason is that when you see the end of our unit at the beginning of May, we only have the lab and lab quiz. That's the only 2 assessments for this unit. Lab quiz, okay? So the lab will be marked very similarly to the, May starts on Saturday, does it? Oh yeah, so that's May 3rd. Oopsies. May 3rd, 4th, 5th, 6th, 7. Thank you very much. Who caught that? Good catch, Colin. I think you answered this already, but can we use props for the video? Yes, of course. Can it be silly? Yes, of course. By all means. Have some fun with it. Yeah, so our unit, we only have 3 weeks of it, and like, you know, 2.5 weeks of this unit basically. And we only have the lab, which will be marked based off of communication like our previous lab. And then the lab quiz, which will be very similar to our previous lab quiz. So this is a very short unit. Normally I have a big assignment designed for this unit, but I think with the timing, I think with the nature of being online, I really just didn't want to give you the assignment. It's a big one. You'd write a 10-page essay on connecting ideas from the first 3 units. And normally we do it at the midterm point for the class, and you know, we're already kind of 3 weeks behind just because of everything that that we've kind of extended, everything in terms of the feedback days, in terms of the breaks being pushed back. So I really wanted to be mindful of this unit as a first unit back from the break, to kind of downshift a little bit and relax a little bit in the context of what we're learning. Because to me, this unit is a fun unit. I love talking about this stuff. I love teaching students about this stuff, and I want you to be excited for it just for the sheer sake of excitement, and not necessarily because you're like, oh, cripes, I got to know patterns of evolution for the test. You still have to know some of the stuff for the lab quiz, but I really want to try to make this unit as relaxing as possible, because the next unit, unit 4, is a little bit more challenging than this unit. So I want to give you a little bit of a breather before that unit. So that's my plan for this unit. We'll finish beginning of May. And my hope is that this unit will give you some time to kind of get back into a groove of school, focus on some units in your other classes, and then you'll have your lab and lab quiz due. And that will be the only 2 assignments that you really have to worry about for this unit. So a nice little break from the norm. Normally we've had our gas, you know, last unit, we really went quickly, I want to say, or not quickly, but it was a lot, right? We did a lot of things over a couple of weeks time. And, you know, I was feeling that a lot of you were mentally exhausted with it. So I really want to try to downshift a little bit. So that's my plan. My hope is that it works out nicely, and my hope is that it's enjoyable for you all, at the very least, to learn about this stuff in the context of just learning. All right, let me do attendance real quick. I will talk about what the plan is for the day. I will go over some key components. You do not have to take any notes. I just want to talk and share some neat ideas with you. You have a couple of videos to watch, and that should all take about 30, 40 minutes, and then you can do some asynchronous stuff, or out of the Zoom stuff, I should say. All right, let's see our attendance. I haven't had to do this in a sweet minute. Oh, also, in the post that I made for this unit, I mentioned some people who reached out to me. My hope is that I emailed everyone back who emailed me. If I missed something over the break and I did not get back to you, please just let me know in the chat. You can just private message me, and then we'll have a quick chat if need be. But I'm pretty sure everyone who got back to me or sent me an email in the break, I've either sent an email back to you on the break or I got back to you in the last 2 days. So if I did miss anyone, please let me know. And I'll be more than happy to chat with you about what it is that you were asking about over the break. All right, Almond is here, Anna's here, Ava, Carson, Colin, Nicole, Daniela, Dean, Emma, Fei Yang, Jessica, Josh, Katie, Krish, Maha, Mariam, Mateo, Matthew, Olivia, Ratika, Rocco, Rose, Sahil, Sarah, Stanley, Sophia, Valeria, Vicky, Bam. Okay, I'm missing someone. Oh, I found them. Awesome. Oh, and to everyone celebrating Ramadan, happy Ramadan to you. I know it was started last week on Monday, I want to say. Monday? Tuesday? Sunset Monday, I think. But to those of you celebrating, happy Ramadan. I know it's one of the bigger holidays for you. So awesome. Hopefully the next 2 weeks will be a nice little break in my class at the very least. I can't promise you for your other classes, but my hope is that you'll find this unit nice. Alrighty. So let's finish talking. You're welcome, Amina. Let's finish talking a little bit about what the plan is for today, because as I alluded to in our Unit 3 outline, I want to kind of recap some stuff from Unit 2 that we're going to connect to. So when you take a look at Unit 2, we did not cover Lesson 10 and Lesson 11. Lesson 10 didn't really connect to any of the things that we were needing for our assessment, but I really strongly encourage you to read through this lesson. There's a lot of neat stuff here. I want to spend some time just talking a little bit about it, because ultimately there's some really cool things with regards to DNA sequencing, coding and non-coding DNA specifically, because if you recall from what we learned, and again, I'm just going over this briefly. You don't have to copy this all down word for word, because I'm really just kind of covering some interesting ideas. This isn't really going to be on your assessment. It's mainly just to kind of set you up for Unit 3. So the one cool thing I wanted to spend some time talking about in this lesson is coding versus non-coding DNA. Most of the DNA that we have is really not that important. Okay, because if you think of the human genome having approximately 3 billion base pairs and 25,000 genes for specific instructions, of that DNA, there's really only about 2% of it that's coded, meaning that 2% of it, of that 2% of those 3 billion base pairs get turned into genes that are expressed as eye color, hair color, skin color, personality, moles, height, weight, all of that stuff. It's really only like 2 to 5% of your DNA. So you're thinking to yourself, well, how can so much variance and so many cool things come out of those 2%? Well, the non-coding DNA, or that junk DNA, the non-gene sequences that do not code for proteins, they are still very important because they help with regards to the cell structure. And what I mean by that is the overall integrity of the cell is dependent on the DNA that is inside of that cell. And so many of those non-coding sequences, like telomeres, for example, when you learn grade 12 genetics next year, you'll learn about something called telomeres. These exist on the ends of your DNA or your chromosomes. And as you age, your telomeres shorten. And it's one of the main theories with regards to aging and stopping aging, quote unquote, is looking at how to elongate those telomeres. Those are the caps at the end of your DNA that kind of protect your DNA from being damaged. And so your telomeres are longer on your DNA than the telomeres in my DNA, simply because you're about 15 to 17 years younger than me. So if you're thinking about getting into a field of genetics, when you learn genetics next year and you learn about telomeres, it'll be a cool little concept for you to cover. Oops. Okay, are there any brief questions with regards to coding versus non-coding DNA? I know it's just a quick overview, but just curious to see. No questions about coding versus non-coding? Does it make sense what I mean by coding versus non-coding? If you could give me a quick little check mark or a yes in the chat. Okay, good. Just making sure it generally makes sense. We don't really, like I said, we're not really going into explicit detail. You'll have plenty of time to do that next year in grade 12 biology if you take it. Who knows? You might be taking it with me in online school, but my hope is that you understand for the most part. All right. Now, the one cool thing with regards to this, and I'm not going to go into detail about it, but I will encourage you to read and I encourage you to watch the video after we finish this lesson, is genetic engineering, because the idea of recombinant DNA, specifically with regards to two or more sources, is a genetic, it's a field in genetics that's really growing and really taking off in the last, 5 to 15 years. So the idea of making changes to the genome of an organism by adding or removing DNA, into a genome, specifically of the human genome, is something that we're becoming more and more familiar with as time goes on. And there's several different ways to do so. I won't go into too much detail. The cool thing about it is for anyone that knows someone with diabetes who needs insulin, the idea of insulin and production of insulin specifically, we essentially hijack a bacteria, insert the DNA responsible for making insulin from the human genome, and then that bacteria then produces that insulin, and then that's purified and removed, and then that insulin can be given to the people with type 1 diabetes, and it allows for them to basically get insulin from an external source. So we have the ability to modify bacteria and viruses to allow for us to take advantage of what they can do, right? Because bacteria grows and divides so fast, we can have them mass produce insulin for us with recombinant DNA. Yeah, so Ritika, the type 1 diabetes that you have that you just told us about, any of the insulin that you use or that you utilize and inject, that is produced by this specific bacteria that is edited, for lack of a better word, with the plasmid or the DNA from the human genome to produce insulin. That insulin is purified once the bacteria grows, and then that insulin is put in a vial and for your use. So it's pretty neat how we can utilize our understanding of DNA, recombinant DNA, bacterial DNA, viral DNA, and take advantage of that to allow for people with type 1 diabetes to to have a somewhat normal life, so to speak, with regards to their ability to have access to insulin. So it's kind of nice. Yeah, it is very cool. So that's one aspect that I just wanted to talk about briefly. And the last one I wanted to talk about with regards to this specific idea is gene therapy. Again, the idea that we can edit sequences of our genome utilizing viruses is pretty neat. It's pretty neat. So I will encourage you to read through it. There's a couple of videos. I just want to make sure you have access to them. I can't remember if you do or not. I'm pretty sure I connect. Yeah, so you can watch some of these videos later after I finish. After I finish going over this lesson and the next lesson, I'm pretty sure. Let me just double check that this one works because I haven't checked since the last semester that I taught it. And yeah, okay, perfect. It works. Oh no, don't work. I don't want you to work right now. I just want you to work in general. Here we go now. Loading forever. There we go. So yeah, I'll give you some time to watch that in a bit. I just want to finish talking about one of the two of the other ideas. You don't have to fill in these questions if you don't want to. It's really just more for you to reflect and think upon as you're watching. So if you want to briefly read through the questions and then kind of think about those questions while you're watching them, that's perfectly fine because I'm not going to collect this. I'm not going to be marking any of it. So you don't really have to worry about filling in these questions. It's more just for your thought because at the end of the day, the biggest thing for me for today is talking about these mutations. Because as we move into evolution starting tomorrow with the lesson properly or with the lesson proper, understanding how some of these mutations work will really allow for us to make those connections. So I am going to talk in more detail with regards to it. I'm not going to have you write down everything. So those of you who are like frantically writing stuff down as I go, it's not going to be detrimental to you if you don't write it down. I really just want to introduce you to the ideas. And then if at the end of my talking in like 10 minutes or so, if you would like to look back at the lesson for this, specifically this lesson, and write some stuff down, that's perfectly fine. I'm not going to stress it right now because like I said, I just want to go over some big ideas and talk about some of those ideas with you as they pertain to what you'll need for this this coming unit. All right. Any questions so far with regards to what I'm talking about? I'll give everyone a minute or two to just ask and okay, gotcha. No, I've already sent in attendance. So you're good. Yeah, you as well. All righty. Okay, so let's talk a little bit about mutation, shall we? The big idea here that I want to spend some time talking about is that mutations are random. Okay, they're random. The ones that stick around are random. They can also happen as a result of choices that we make, which are called induced mutations. So random DNA errors are just errors in DNA replication. We talked a little bit about it in last unit when we looked at mitosis. At that S1, that synthesis phase, when DNA is being copied, and we look at the end of S1, if there are DNA errors that stay around, those are what's called random mutations. Okay, random mutations. Now, the induced mutations can be caused by environments. Smoking, for example, any type of harsh drug, alcohol abuse, substance abuse in general, radiation, whether it be ultraviolet, nuclear, anything of that sort, microwave, all of those can be what's called induced mutations. Now, the key thing here is that our cells really try to fight off these mutations. If it sees a mutation, it goes through that apoptosis, right? It's really trying to avoid that random mutation or those induced mutations because those mutations can lead to cancerous cells, and those cells can have negative ramifications for us if they're allowed to kind of propagate onward. So there's two main types of mutations within the DNA, just the DNA itself. And then I'll talk a little bit about chromosomal mutations, but I just want to talk a little bit about the two main types of DNA mutations for a bit. So the first one is called a point mutation. A point mutation. This is a single base error. A single base error. So a C turns to a G. A T is inserted. And then, or a C is deleted. All of those are point mutations. So it can be either a substitution, where one specific base is changed to another one by accident. There could be an insertion where one is popped in, or there could be a deletion where one is removed. So the words are, for the most part, pretty self-explanatory, I hope, that if one is substituted, it's just changing. You insert one in between, or you can delete one and remove. The second is called a chromosomal mutation. And there are multiple different types of chromosomal mutations. We won't focus on it too much, simply because we don't go into huge details in the next unit with regards to chromosomal mutations. But I just want to cover some of the basics of it, because it is quite fascinating to consider that whole chunks of chromosomes can be either deleted, added in, removed, or swapped. And it's important to remember that, yes, this chromosomal swapping is called crossing over under the right circumstances, because a mutation, and this is what I wanted to kind of focus on today, a mutation involves that large piece, a chromosomal mutation involves that large piece or whole chromosome. But it's normally with regards to either an alteration in the chromosome structure or a change in the chromosomal number through non-disjunction. So we've looked at non-disjunction before. We've looked at alterations in chromosome structure between those homologous chromosomes, but a chromosome mutation is either between a non-homologous chromosome or a non-disjunction disorder that happens as a result of a change in the number. So we've kind of seen that already. So if a non-disjunction disorder happens, right, trisomy 21, for example, we consider that a chromosomal mutation. Okay, so two main mutations. I'm going to talk a little bit about the chromosomal mutations. Are there any questions about either of these mutations right now? Everyone's okay with the general idea of the two types of mutations that I've talked about so far? I'm going to go into a little bit specifics with chromosomal mutations, but again, I'm not expecting you to memorize this. It's not going to be assessed. It's mainly just to have you connect to some of the ideas we learned in Unit 3. Is it possible for two of those to take place at the same time, like A is deleted and C is substituted? It's very unlikely. We will talk about the percentage chance for all of these to kind of happen when we start our next unit. But like the chance of a mutation sticking and for it to be passed on is like 1%, if that, not even, like 0.5%. So it's very unlikely for two of them to happen at the same time like that. Unlikely. I didn't say it's impossible. It's just unlikely. Okay. Cool. So let's take a look at some of the mutations with regards to alteration in chromosome number and chromosome structure. The first is called a translocation. This happens when a whole piece of the chromosome moves to a non-homologous chromosome. So if you can imagine, we have two chromosomes here. Number 2 and number 15. What translocation is a chunk of this, number 15, goes over to number 2. It's not the same as crossing over, because remember, crossing over happens between homologous chromosomes. This is between non-homologous chromosomes. So there is potential for non-homologous chromosomes to have DNA kind of swap or move from one chromosome to the other that in non-homologous pairs. So this is bad, right? This is bad. Usually if this happens, this mutation does not allow for that cell to continue to survive. Usually. Again, I say usually because I'm really trying to prime you for the idea of next unit where we look at some mutations sticking around because they end up being beneficial. And it's neat to consider that normally 99.9% of the time, any of these mutations we look at, if it happens, say goodbye. That cell or that organism won't make it to term, or won't survive, and that's the end of that chapter. Literally billions upon billions of mutations happen daily on this planet in species, and maybe only about 100 of them end up sticking around. And of those 100, maybe like 2 of them are actually helpful. So if you've had dreams of mutating into like a mutant from like X-Men or something, chances are very, very low for you on that one. So translocation, it's when non-homologous chromosomes kind of switch or pop over a piece of their DNA. Deletion is exactly what it sounds like. Oh, question. If part of 15 goes to 2, then does part of 2 also go to 15? No. So what ends up happening is that this part here that I've highlighted, used to once be here. So that's gone now, though. It's gone. It's cut down 15 and increased the size of 2. All right. So let's take a look at some of the other mutations. They're a little bit more straightforward in their name because a deletion occurs when a piece of the chromosome is deleted, right? I've started numbering it in terms of 1, 2, and 3. And as you can see, before the mutation happens, we have a section with 1, 2, then 3. And as you can see here, 1 is gone and we're only left with 2 and 3. So that's where there's a deletion. It can be harmful or it can have no effect at all. It really depends on where that section is deleted. It's all contextual in terms of where that section is deleted. If it's a real big chunk that's deleted in like an important sequence that codes for brain development, arm development, whatever, then you can start to lead to harmful mutations. Or it could be a section of DNA that has absolutely no importance at all, and it won't have an effect. All right. So the duplication, as you can imagine, duplication is when a section is duplicated. So here we have a repeat of section 2. Let me highlight that. Because it was duplicated by accident. And if it's only repeated once or twice, again, usually it has no effect. It really has an effect if there's way more than 2 duplications, or if there is a specific part that is duplicated. That's very important. So again, very contextual. if you only have one or two tiny duplication events on a chromosome or two, chances are it really won't impact the outcome of that organism. Once it starts to be harmful is when you have several duplications on a chromosome or several important duplications that happen. And then the last thing is the inversion mutation. This can be one of the most insane mutations out there because you can start to see so many different things happen as a result. And I kind of talk a little bit about it with regards to sickle cell, but we'll lead into that in a bit once I kind of go over the whole idea. Okay, so what happens here in an inversion is that the reversal of fragment order. So it's not that one gets deleted, multiplied, or copied, or removed altogether or added in. This is where we see the order change. Okay, we see the order change. So instead of it being the 1, 2, 3 that we have here, we see it being 2, 1, 3, or the 1, 2, 3 flips to the other side of the chromosome and it becomes 3, 2, 1. Those are all inversions. It's just changing the order of the DNA. And in some body cells, this can be quite bad because the order with which genes need to be read is usually pretty important. These DNA mutations won't normally be passed on. And it's, again, the key thing here is that only the mutations that happen in gametes are going to be passed on. This is crucial for us in this unit because evolution is based upon the idea that these mutations happen in the gametes and not just in the cells, like the somatic body cells. All righty. Any questions about any of these mutations before I really connect it to everything that we've learned? Because this next part, I will tell you now, this is the big one. The most important thing that we have learned so far in this class, we are going to connect all of those ideas to this concept and then how it will connect to evolution. So my hope is that if you have some questions about anything really over the last unit, 2 units, 3 units even, let me know and I'm happy to talk about it because I'm going to make one example connection and then I'll talk about what's called the central dogma of biology, which is kind of the big one, so to speak, that connects everything we've learned so far. Judging by the silence, I'm assuming that everyone's okay. Judging by the 2 check marks that I've gotten, everyone's okay. First day back, everyone's a little bit slow. That's okay. I know that this is a lot of information. Again, it's not meant to be memorized. It's not meant to be like, oh, dear God, I have to know this. It's meant to just be like, oh, that's neat. That's cool how it works. I had some thoughts about it. Was eating breakfast, couldn't type. That's okay. All right, cool. So let me introduce you to a specific example. And that specific example we've kind of talked about already, and that is antibiotic resistance. Antibiotic resistance happens when a specific bacteria becomes resistant against a specific antibiotic. And this can happen as a result of a random mutation. So recall, bacteria replicate millions upon millions upon millions in the course of a day. If one of those cells has a random mutation, and one of those cells' random mutations is in the DNA responsible for making the cell wall, and it creates a peptidoglycan layer that is slightly thinner than what the antibiotic needs in order to work, that bacteria will become resistant to that antibiotic? For a point mutation where a letter changes, is that counter letter change too? Yes, it does. It does. And this is for DNA and RNA. Any type of genetic material, DNA or RNA. But yes, that's a very important question, Matthew. Matthew asked, if a point mutation happens with one strand, does the counter letter strand change as well? Yes, it needs to. Because remember, those DNA molecules won't connect unless the correct opposite base is opposite of it, right? All right, so that antibiotic resistant example I was talking about, this is one of those times where this mutation is beneficial to the bacteria, but extremely harmful to us. So that bacteria that's now resistant as a result of a single tiny point mutation or a single chromosomal mutation, now that has changed the way that bacteria behaves, it changes the way that it responds to antibiotics. And so now we can no longer treat these bacteria with the medications that we've discovered. So a specific bacteria, the specific bacteria is called MRSA. If you're curious about it, it's a methylo-resistant Staphora. Oops. I did the short form version of it. You can take a look up. You can take a look. Wow, again, this is my first time speaking like this in a long time. You can take a look at it if you would like to. It's quite interesting to look up. If you just type in MRSA, you'll be able to learn a little bit more information. But this MRSA is a type of Staphylococcus aureus that went through a mutation that prevents it from being broken down by the antibiotics that we have. And it's a skin or a tissue bacteria that grows. And if it grows out of control, it can actually be quite harmful and deadly to humans. And so far, we are unable to treat it with bacterial antibiotics. So this is an important example to kind of consider because, and again, I'm just giving this as a, screen, where'd you go? I'm just giving this as an example to kind of connect it to all of the idea that I'm going to speak of to next. Because when we consider how DNA works and why it allows for things like this to happen, we must consider, I'll do it two different chunks. We must consider what's called the central dogma of biology. Now, the central dogma of biology is the overall idea that we as biology students and biology teachers and biologists as a whole, we must prescribe to the idea of the central dogma of DNA in order to make the connections that we've learned to everything that you can learn in biology. So when I ask the question, how do mutations have such serious effects? Why is a genetic mutation so important to consider? It's the central dogma, the idea that DNA is transcripted or turned into RNA, which then gets translated by the cell into proteins that makes our traits possible. The movement from DNA to RNA to protein, which is the central dogma of biology, we accept this idea that DNA gets made into a protein that we then utilize in a cell to give us blue eyes, red hair, lighter brown skin, darker brown skin. All of those traits that we as a species and as living organisms are made up of, it's all because of the central dogma, the idea that DNA turns into proteins. And then those proteins create amino acid sequences, or those proteins are made of amino acid sequences. They fold, they behave in a special way. So if there is an issue with one of those sequences of DNA, another example that we'll use is sickle cell anemia, a normal DNA sequence that produces a very specific protein structure that allows for blood cells to have that normal biconcave shape. If we see a mutation in this middle sequence, which creates a mutation in the RNA, which creates a different protein than what your body needs to make a healthy blood cell, you see that this can change the way that the protein behaves, which changes the way that the blood cell shape works, which ultimately changes the way that that functions. And so this central dogma, this overall idea that from DNA to RNA to protein, this is the overall idea that we must accept or at least prescribe to in order to make the connections that we've learned with regards to our first 3 units and really the next 2 units or next 3 units as well. So the pro, val, glut, these are just proteins. You don't need to worry too much about them. You haven't learned about protein growth or development yet. These are all proteins. You'll learn more about it in grade 12. But for just the sake of central dogma concepts, the idea that the DNA gets turned into proteins is an important one to understand. All right, questions, thoughts, ideas, concerns. Because you'll see this tomorrow when we start our first lesson in Unit 3, how it all kind of connects. We will connect the idea of this central dogma and the overall idea of genetics to evolution on a wild ride that will last for 3 weeks and hopefully make it an enjoyable learning experience for you. All right, folks. So you have a couple of videos to watch. If you want to read through the note one more time on your own, by all means, I will post this lesson later on today in case you want to rewatch it to provide some more context for you. But it's 9:55 now. If you would like to leave the Zoom, take a bit of a breather, read some notes. If you want to stick around and talk to me, ask some questions. If you want to work on your project, I'm giving you this time from now until the end of class to do so. So if you want to stick around for that, I support that. If you want to head out of the Zoom, I'll still be here to answer questions. And yeah, good first day back. A little bit of a relaxing one, I hope. Not too heavy, just a little brief connection to what we're going to learn or what we're going to use from our previous unit to what we're going to need for our next unit. So if you're heading out of the Zoom, work hard. And I'll see you all either in Zoom later today if you have questions or tomorrow. You're welcome, folks. Have a good one. Enjoy your first day back, folks. Bye, everyone. Work hard.
Bye, Mr. Kitasol. Have a great day.
Thanks. You as well, Matthew. Take care. Can it be two separate videos? And can we share our screen and talk like that? Sure, that's okay, Jessica, 100%. As long as I can see your face, it can be two videos and you can share your screen as well. I'm perfectly fine with that. Yeah, no worries. When it comes to terms we can and can't use, can we use Punnett square? Or would I have to say like a complicated tic-tac-toe board or something? Say complicated tic-tac-toe board or multiplication chart. How do we know the terms that we can't use? Okay, so the best way that I'm helping students to know what they can and can't use, if I taught you the word, don't use it. So when I taught you about Punnett squares, that's probably the first time that you learned it. Don't use Punnett squares. Okay? Yes, that's the whole challenge. So any term we made to explain in further detail, don't use. Correct. If you say Punnett square and you have to tell me what a Punnett square is, then you probably shouldn't use Punnett square. Yeah, I hope you had a nice break too, Emma. See you tomorrow.